Good morning, everyone. I'm happy to see everybody again. I just wanted to apologize a little bit. I know I fell a day behind this week. I've been juggling a little bit because I don't know if you probably don't know, but our school's going to be closed on Friday. We have a mandatory data summit that we have to attend. And so what that means is I have to juggle a whole lot of things, including media that I have to make for my face-to-face and things like this. So, well, there's only so many hours in the day. And a few of you got worried because the quiz was late a day. And anyways, I want to apologize for that, okay? Hopefully it won't happen. I don't foresee it happening again this term. This is the only hiccup that we've had. So enough about that. Let's talk about this chapter. Okay, this is chapter 10, and we're talking about controlling microbial growth with antibiotics. This is one of my favorite chapters because, again, as a biochemist, I'm a person that likes to know how things work. And this chapter does a wonderful job of explaining how the main mechanisms in which these antibiotics function. So one of the first things that we need to introduce, which I'm sure you already know, is the fact that the source of antibiotics are other microorganisms. Fungi were originally the main sources of antibiotics, and then recently has been other bacteria. So as logic dictates, bacteria have to defend themselves against other bacteria. So consequently, evolutionarily, they would have the best way of defending themselves, and we harvest those defenses as antibiotics. Okay. This slide is important to me because this explains the main motifs, the main mechanisms by which antibiotics function. Okay. So starting over here, you can see that one of the main ways is to inhibit attachment of the microorganism to the host tissue or the host cells. We've made the argument multiple times that if you can inhibit the attachment of the microorganism, you have effectively inhibited its pathology. Okay, so that's very important. We might inhibit the production, the synthesis or production of DNA and RNA. That stops the central dogma from the beginning. And by central dogma, I mean DNA to RNA to protein. So you cannot make the proteins necessary for life if you cannot synthesize the nucleic acids that are DNA and RNA. So that is one way to stop microbial growth. Another one is to stop an essential metabolic pathway, okay? And this is typically done by sulfa drugs. So this is why sulfa drugs are incredibly powerful, because they stop very essential metabolic pathways. And another thing they should know is that broad spectrum antibiotics typically function by inhibiting an essential metabolic pathway. So if you're looking at the big guns or the most powerful sulfa drugs, we're stopping some essential aspect of the microorganism. We have disruption of cytoplasmic membranes. This is not typical at all. This is something that's a little bit rare. However, it's important in the defense against fungi. So, with bacteria, not so much. And this is used rarely because, again, it doesn't function against many microorganisms. But the microorganisms against which it does function, we typically don't have other defenses against. So this makes it very important, even though it's used rarely. Inhibition of protein synthesis. So this targets the ribosome. So again, in the central dogma, we have DNA to messenger RNA to protein. The ribosome is essential for the synthesis of amino acids into proteins. So if you can arrest the function of ribosomes you're inhibiting the production of essential proteins, and you're shutting down the microorganism. One of the most best motifs or mechanisms is actually the inhibition of the buildup of the peptidoglycan cell wall. So this is particularly important for gram-positive microorganisms that are known to have a thick peptidoglycan cell wall. Gram-negative microorganisms, which have a thinner peptidoglycan cell wall, are also susceptible to this, but not as much as gram-positive. So if you are fighting Staphylococcus aureus, this is the main antibiotic mechanism that you're going to use, is something that inhibits the production of that thick peptidoglycan cell wall. Okay, so staying on the same topic of that cell wall in gram-positive organisms, hopefully it's review for you to remember that peptidoglycan is made out of two sugars, two carbohydrates, NAG and NAM. So N-acetylglucosamide and N-acetylmuramic acid. You don't need to know those. You can just remember them by their acronyms, NAG and NAM, okay? And the cell wall, so those are effectively the bricks of the wall. Now what we have to do is we have to mortar them. We have to put the cement between the bricks in order for that cell wall to form, okay? Those are called cross linkages. So if we can prevent the cross linkage between a NAG and a NAM, we're effectively building a wall of loose bricks that is not going to stand around. So that is a way that the majority of antibiotics that target the peptidoglycan cell wall, that is the way that they work, is by targeting this cross linkage. This is the most prevalent, this is the most common way of undermining the production of this wall, okay? How did this happen? Well, all these antibiotics have this little this carbon ring. Do you see it here? It's called a beta-lactam ring, and it consists of 1, 2, 3 carbons and a nitrogen. And I gotta tell you, carbon doesn't like to be in that square. It doesn't like it. If you've taken any chemistry, you'll remember that carbon likes to be in what's called a tetrahedral shape, which is the angles are somewhere around 109 degrees. So if you take the angles of carbon, which are supposed to be 109 degrees, and you bend them to form a square, like in this beta-lactam ring, then there's ring strain, and the carbon is not happy, okay? So the metaphor that I use is that I say that it's like having a little mouse trap, and you force the little hinge with a spring, right? And then you set your trap, and that trap is ready to catch something. Well, that's what this beta-lactam ring is. You take carbon and you force it into a square, and it's not happy. It wants to explode out of that square. Okay, so what happens? Now the cell that's building the wall, okay, accidentally incorporates this antibiotic in there. And as soon as the cell carries out chemistry with this antibiotic, that little square explodes and it breaks the cross linkages between the NAG and the NAM, consequently producing a wall that is not being held together, okay? So you can see that all these antibiotics that target the peptidoglycan cell wall has that beta-lactam ring. Do you see that? Whether it's the original penicillin or methicillin or some of the more contemporary ones, they'll all have that same general motif. So what happens? Now that the cell wall has been undermined and those cross linkages are no longer there, now the cell cannot protect against the osmotic pressures of being in a hypertonic environment. And consequently, osmotic pressures take over Okay, what does that mean? If it's in a hypertonic environment, water is going to want to come out of the cell to the outside because that peptidoglycan cell wall is not preventing that anymore, and you end up with a lysis of the cell. Okay, so what do we end up? We break the cell wall, and then osmotic pressures take over and lyse the cell. Okay, moving to another one. Okay, now we're talking about disrupting not peptidoglycan cell walls, but disrupting mycolic acid cell walls. Okay, and what are we talking about? We're talking about mycobacteria. What are mycobacteria? Well, mycobacteria are worse. They are worse than gram-positive organisms. Okay, because gram-positive have a thick peptidoglycan cell wall. Mycobacteria have a thick, waxy wall that is incredibly difficult to penetrate. Okay. Furthermore, their metabolism is typically very, very, very slow. So we are talking about tuberculosis. We're talking about leprosy. Okay, those are mycobacteria. And so we have this isoniazid and ethambutol, okay? And what they do is that they disrupt the synthesis of this mycolic acid, this waxy wall, and it makes these mycobacteria susceptible to the environment, okay? This is very specific, but then again, we're targeting a very specific type of an organism, a mycobacteria. Okay, it's in red and is in capital letters, so it must be important, right? As I said at the beginning, most broad spectrum antibiotics function by inhibition of protein synthesis. In other words, the most powerful and far-reaching antibiotics inhibit the production of protein, okay? And look at this. There's so many motifs. This is beautiful, okay? We're talking about 6 different ways in which this can happen. So here we have aminoglycosides, okay? Let me enlarge it for you so you can see it. So what are we talking about? Streptomycin, for example. What does it do? Notice that all these target the ribosome, okay? So streptomycin, what it does is it associates with a small ribosomal subunit. It changes its shape. When it does that, it messes up the organization of the codon with the anticodon of the transfer RNA. They don't match very well, and so the wrong amino acid gets incorporated. Do we care? Yeah, we care, because now this microorganism is synthesizing a garbage protein that is not going to be useful to it. So that is one example. Here's a second one, tetracycline. I don't know how many people have been on tetracycline. We're coming off it a little bit because apparently it gets incorporated into the teeth and it causes problems. So we don't use it as much as we used to. But look at its function. Tetracycline literally docks on the A site of the ribosome. If you remember, this is an A site, the acyl, the P site, the peptide site, and the E site. And what it does is it associates with the A site, stopping the association of the tRNA anticodon with the codon of the messenger RNA. That's it, we're done. No protein is going to be synthesized. Fantastic. Let's look at chloramphenicol. So chloramphenicol, as you can see, is associating between one amino acid of one tRNA and the amino acid of another tRNA. And what it's doing is preventing the synthesis of the peptide bond between one amino acid and the next. So again, when this is done, the amino acids come out, but they're not a protein because they're not linked together. That's fantastic. Let's look at another motif. Okay, these are macrolides. I'm fascinated by macrolides because they're incredibly large antibiotics. As a matter of fact, the name macro means large, okay? So these are very large antibiotics, You can see what they do is they tether the messenger RNA with a large ribosomal subunit, and in doing so, it prevents the ribosome from translocating to the next codon. So that ribosome cannot move from one codon to the next. Again, we have stopped protein synthesis. Here's one that a lot of people are excited about. I am not too crazy about. This is short interference RNA. And short interference RNA is a small piece of RNA that interferes with translation by associating complementary with a messenger RNA. And the reason I'm not crazy about it is because in order for this guy to work, it has to be in the cytosol of the cell. And as I have said before, our cells are not, they're not kind to genetic material in the cytosol. If they find messenger RNA in the cytosol, they'll degrade it very, very quickly. So the half-life of the short interference RNA is incredibly short. Here's oxazolidinone. I can't even pronounce it anymore. Oxazolidinone. I should practice that some more. But what does it do? It associates with the messenger RNA, and it inhibits the production of the initiation complex of translation. Fantastic. We can't even get started. So we don't have to worry about whether or not we're going to make a protein. So these are 6 mechanisms in which we can stop the production of protein. And this is a great way of inhibiting bacterial growth. Now we're going to look at membranes. Okay. So now we're not looking at the inhibition of proteins. Now we're looking at a different mechanism. And what we're going to do is we're going to disrupt cytoplasmic membranes. Effectively, what we're going to do is we're going to cause holes to appear in them. Okay. So here we have amphotericin B. And what this guy does is it attaches to a cholesterol molecule in fungi. Okay. So here's a picture of it. Here's a cholesterol molecule in the fungi. It's called ergosterol. This amphotericin B associates with it, and they'll do it on the inside as well, and then they'll move independently in the outside and inside leaf of the plasma membrane until it finds itself. And once it finds itself, it locks up like this, and it creates a pore. Well, you can't have a plasma membrane with a hole in it. If you do, everything that's essential inside is going to escape to the outside, and everything that you don't want coming in is going to come to the inside. So effectively, you have just lysed this fungal cell. And this is very important because we do not have a lot of antibiotics against fungals or fungi. So this consequently becomes very, very important. Okay, let's keep on going here. Here we go. So the next important topic is inhibition of metabolic pathways. And this is where sulfa drugs come in. So if you've ever been in a clinic and they're running short on antibiotics, the clinician is always going to make sure that the sulfa drugs are well stocked. Okay, and what they do is they inhibit a metabolic pathway. So here's an example of that. Okay, here's a pathway where we start with PABA. PABA stands for para amino benzoic acid. Effectively, this guy right here. Okay, and we go through a whole bunch of enzymes in this metabolic pathway to create the precursors necessary for, not surprisingly, DNA and RNA. Okay, which enzyme are we affecting? The very first one. This is arresting the entire metabolic pathway. If you shut down this enzyme, nothing downstream is going to be synthesized, including DNA and RNA. That's a good way to stop a microorganism. So how do we do this? What we do is we create sulfonamides, okay? So sulfa means that it has a sulfur, and amide means that it has an amino group, a nitrogen. There's my sulfur, there's my nitrogen. Sulfur, nitrogen, sulfur, nitrogen. Sulfonamides. Okay, now what? Well, it turns out that this benzene ring, this aromatic ring, and this amino group are identical to the ones in PABA. So that means that these sulfonamides are analogs of PABA and can compete for that active site in that primary enzyme, okay? So compete, what does that mean? That means that sometimes, part of the time, the PABA is making in there and the system is going along, and part of the time, an analog is going in there and not doing anything, okay? So at the very least, it slows down the synthesis of DNA and RNA. But some of these larger sulfonamides do more than that. They don't just compete for the active site. They enter the active site and they stay associated with the active site, rendering that enzyme non-functional. That's fantastic. Because now that means that from that point on, that enzyme is not going to produce any of the precursors of DNA and RNA. Okay. So as a role of sulfonamides, okay, let's keep on going here. Now we're looking at the use of nucleotide analogs, okay. Nucleotide analogs are used based on two criteria. One is that you have an organism that is reproducing very, very, very quickly. And that's important because that means that it will assimilate the analog more quickly because the metabolism is much higher. The second one is that it's kind of a Hail Mary. You are desperate, you need a solution, and this might be the only solution that we have, okay? So what would replicate very, very quickly? Well, viruses is a good example. Cancer, one of the hallmarks of cancer is the fact that cancer cells grow and divide without any impediment, okay? So that means that these nucleotides would be associated with these viruses and these cancer cells more often than it would be with our cells. And consequently, they would introduce mutations into these viruses and these cancer cells at a much higher rate than it would in our cells and undermine the viability of these viruses and these cancer cells. So this is what these nucleotides look like. We have analogs of all four of them. Here's adenosine, here's the analogs. Here's guanosine, here's the analogs. Here's cytidine, here's the analogs. And here's AZT. AZT is one of the known analogs that we use in the cocktails for HIV patients. And that's an analog of thymidine. Very, very useful. Okay, let's keep on going here. Reverse transcriptase or inhibitors are very important because if you remember, you have DNA, messenger RNA, protein. To go from messenger RNA back to DNA, you need to go in reverse of transcription. The enzyme that does that is called reverse transcriptase, and that is only carried by retroviruses. Viruses with genetic material is RNA, okay? So if you can take a reverse transcriptase inhibitor that will only affect the reproduction of retroviruses, and it will not affect us in any way. So these are fantastic treatments. Let's see. Narrow and broad, well, here's erythromycin, tetracycline, sulfonamides. See how broad those are? This implies that they are stopping a metabolic pathway. These are a little bit more specific, but as I told you, that doesn't make them less important because the very specific ones typically target microorganisms that we are typically not able to stop in any other way. Okay. What else do we need to know? Here's a Kirby-Bauer test. I believe it's one of the things that we're going to be doing. And this is how we test antibiotic susceptibility. What you do is you take a Petri dish, you make a carpet of the microorganism. What microorganism? The one that we got from the throat culture, the one that we got from the urethral culture, whatever. You make a carpet of it, and then you put different antibiotics. There's tetracycline. So it tells you the antibiotic, and it tells you the concentration. And you can see these zones of inhibition. Okay, those zones of inhibition allow us to use a interpretation chart and discern whether the microorganism is susceptible or not susceptible to each antibiotic. Okay, so hopefully we'll be doing this. Okay, and just two more things I wanted to tell you as before we finish this chapter. One is the role of efflux pumps. Okay, one example of an organism that uses efflux pumps is the opportunistic microorganism Pseudomonas aeruginosa. Okay, Pseudomonas can actually grow in a vial containing antibiotics, which is very scary. And the way they can do this is by using efflux pumps. In other words, it takes the antibiotic as it diffuses into the cell and it pumps it out. And so the antibiotic doesn't get an opportunity to do anything. So this is one of the scariest outcomes. Pseudomonas aeruginosa is an opportunistic infection of burn victims. And you can tell that when it's taking place because the purulent disposition on the surface of the burn victim becomes green, and that greenish color is the pyocyanin, the dye being released by the Pseudomonas aeruginosa. And why do we care? Because these efflux pumps and the Pseudomonas aeruginosa makes it particularly challenging to treat with antibiotics. How are you going to treat something with antibiotics when it proactively pumps antibiotics out of the cell? It gets challenging. And then the last topic, as we finish, is the understanding of synergism. Okay, so if somebody finds himself in a hospital and they have a challenging infection, the clinician will probably put them on 2 or 3 different antibiotics simultaneously. Okay, why? Well, here's a carpet, like in a Kirby-Bauer plate. Here's one antibiotic. Here's the other antibiotic. And look at what's going on in the middle. There's a large zone of inhibition. This large zone of inhibition is where small amounts of this antibiotic and this antibiotic have diffused together and worked synergistically to destroy the organism. And synergism implies that the whole is greater than the sum of the parts. In other words, the effect is multiplicative. So it's a lot more powerful than either one by itself. So synergism, the cooperation in the activity of multiple antibiotics, is a very important clinical point. Okay, so that's this chapter. I'll come back in a little bit and we'll address the next one. Okay, Have a wonderful morning.